Imagine we're inside an E. coli cell. I'm the cell's DNA, so I have all the instructions we need to run this whole operation in my book right here. The people behind me walking around on their cell phones completely oblivious to the world around them, they're ribosomes. They're pretty dumb, but they're great at building the proteins this cell needs to survive, and they'll pretty much do anything that I put in our massive cell-wide group text. Hey everyone! Let's start making lactase!
Great idea!
The problem is, they'll literally do anything that ends up in the group text, even things put there by viruses.
Hey everyone! Let's make viruses and then kill each other!
Great idea!
Luckily, we have this guy named Cas9 who carries around a giant pair of scissors and cuts up viral messages before the ribosomes even know about it. The problem is, those viral messages are made of DNA, just like my whole book here. Luckily, Cas9's a pretty sharp guy. I gave him a strand of RNA, which is a text fragment only viruses would send out. He'll only cut messages with that text fragment, letting my book of DNA go untouched. Unfortunately, viruses don't always send out the same message. That's why we have more than one Cas9 protein. They're exact copies of each other, except I give each one a different RNA strand to look for, which means they look for different text fragments. If Cas9 doesn't recognize a virus, a small piece of identifying text is captured and stored in a special chapter of my book called the CRISPR Locus.
The CRISPR Locus stores all Cas9 text fragments as spacer DNA between short repeating sections of palindromic DNA. These sections are also known as CRISPR DNA because they're clustered, regularly interspaced, short palindromic repeats.
Under normal circumstances, Cas9 will never cut a cell's genome. But what if we wanted that to happen?
Behold, the Human Genome, a book that contains all the great things that make you human, like having blue eyes or having lungs, but sometimes you might end up with a less than ideal trait, like having only 4 fingers. Let's zoom in on that. Scientists discovered that it's possible to hijack Cas9 proteins with specially manufactured RNA text fragments. These Cas9 proteins can then be injected into a cell to cut out an unwanted trait. To complete the edit, all that's needed is a new, slightly different strand of DNA provided by the scientists. Despite the differences, it's incorporated into the cell's genome anyway. Now, if this procedure was performed in a fertilized egg cell, every new cell would have the same change, creating a human with the new trait. Using this strategy, the CRISPR Cas9 system could help treat genetic diseases like muscular dystrophy and cystic fibrosis, and could even be used on other organisms.
But before we get too carried away, make sure you thank your local bacteria for the CRISPR Cas9 system.